So, final talk of day one, Vadim Kaiser coming to us from Sac State for now. Talking on phenomena, artifacts, and effects in biological measurement.
Cool. Thanks, y'all. So this one's got some aliens in it, and this is no joke. So introduction, we talk about artifacts. We use the word artifacts quite a bit, and artifacts, or just generally results that are products of error, are everywhere. And a lot of times we actually see this in consistent results. So we'll have results that seem like they are robust, and I'll explain that word in a bit. But actually there is systematic error that's going on underneath them. One example is faster than light neutrinos. I know you all probably heard about this. This one was very exciting, 2013. So the details, you had physicists at CERN, and they found that these neutrinos traveled 60.7 nanoseconds faster than the speed of light. It was very exciting, specifically because there was careful measurement that was involved. That was the first thing that I looked at, is how careful is the measurement. So time was measured with a GPS timing signal, of course. There were small movements that were being tracked in the Earth, factors such as day versus night, seasonal trends, proton pulses. This is a really good one. The initial criticism is maybe it's something with the proton pulses, so they were varied to make them shorter, longer. And what's amazing about this is this was replicated 15,000 times. So this was something that was, like hyper robust. It was like big data robust. But the problem was that there was an error that was involved, which was that they didn't really plug in the fiber optic cable so well. So there was a loose connection. Once they plugged it in, it took away the results, and these neutrinos did not travel faster than the speed of light. So each trial had this systematic error. So another example, in the form of self-analysis, we do this all the time. This one's kind of a cool one. There was a guy named MacArthur Wheeler who robbed not one bank, but actually two banks, and then the police catch him about an hour and a half later sitting watching some television, and he's very surprised. So he goes, how'd you find me? And they go, well, you didn't wear a mask. And so he starts shouting at them. He goes, I wore the juice. I was wearing the juice. And they're like, what juice is he talking about? Is there something, what's going on with him? Is it perhaps a mental health condition, substance? Something's going on. In fact, no, he went through a psychological evaluation. He was completely fine. In fact, he was convinced that he had this theory. He was convinced that it was corroborated that putting lemon juice on your face makes you invisible to cameras, and it sounds pretty silly, but he used multiple means of measurement to test this out. He had a Polaroid camera and a camcorder, so he squirted the lemon juice in his face and he recorded himself, and he didn't appear in the after image. And the reason why was because he pointed the measurement apparatuses away from him. I don't know if you've ever tried putting lemon juice in your face, like you've tried one of those new age lemon juice cleanses or whatever. So each technique was compromised by what social psychologists call the Dunning-Kruger effect, which is when you're too stupid to know that you're stupid. So your incompetence masks your incompetence. We see this all the time on, it's like American Idol, and people think they can sing really well, but they can't, and they can't understand that they can't. So consistent results can be compromised. And this one, this right here is apparent in a lot of scientific research, but I want to talk about inconsistent results. So I'd like to talk about a very specific subset of scientific practice, which has to do when results are not consistent or not invariant or not robust. So I'm going to talk about intervention-based regularities, and what I'm going to try to discover in this conversation, I want it to be more of a conversation that extends into the future, is I think that intervention-based regularities are neither artifacts nor phenomena, natural objects, and I'll explain what those are. And characterizing them as such fails to take into account their special purpose in experimentation and also what we can learn from them. So the outline, gone through the discussion, I'm going to talk about artifacts, the puzzle of variant results, phenomena versus effect, and intervention-based effects, which I call context-sensitive effects or unstable effects sometimes, variant effects. So let's characterize artifacts. How do we differentiate genuine results from artifacts? Well, there's a great tool that we have in philosophy of science called Robustness Analysis, and this right here allows us to understand when a result is produced in error. So a really simple characterization of this, you use multiple means of information gathering in order to converge on a result. So according to WIMSAT, it differentiates real objects, events, and processes from artifacts, which are results produced in error. So we do this all the time perceptually. You might see a mirage. Ellie doesn't ever do this perceptually. He just has direct perception of reality. So I'm going to point at you with the laser pointer. So we do this all the time. You see something that you might think this is a mirage. You're perhaps sleep deprived. This is one of those things. It's finals week. You've been doing a lot of grading. You might start to see some things. So you go and you touch them. This is a form of robustness analysis. You reach out with a different mode of measurement. If all of your modes of measurement are producing the hallucination, then you are in trouble, and that is probably not the greatest thing. But this is what we do all the time. So we can characterize artifacts using this characterization. So according to WIMSAT, the characteristic of artifactual results is that they are unstable in the context of multiple independent methods of identification. So these results are unstable, and when we have results that are unstable, we think that they are unreliable. This is the inference that we make. I'm going to see if maybe we can shift our perspective on this a little bit. So examples of artifacts, we have artifacts in cellular structures. I'm sure people have heard about this. If you observe a cellular structure, you see all kinds of things going on. For the most part, appearances seem like they are straightforward, except for when you're talking about things like the mesosome. There's been a large debate in the philosophy of science, history of science literature on the mesosome. Appears to be a genuine cellular structure in many different appearances written about in textbooks. I remember seeing the mesosome in my biology textbooks. People posited the function of the mesosome. However, Rasmussen talks about, and this is kind of a controversial case study, but he talks about the fact that a mesosome is just an artifact. How do we know this? By varying staining procedures. So when we vary staining procedures, we see that certain ones produce the mesosome, other ones do not produce the mesosome. So what you have is an unstable thing that is being produced, and so you make the inference that this is not a genuine cellular structure. Let's talk about aliens. Okay, so this is where we get into some alien science, and I'm going to get kind of far out. So Mono Lake is a very mysterious place, and in fact, when I started, I just started Googling pictures of Mono Lake, and the first thing that comes up is I guess they found a Sasquatch at Mono Lake. That was like the initial thing. That's the proper scientific name and pronunciation, the Sasquatch. So I was thinking, it's a very mysterious place, and they found, I remember reading about this also, they found this microbe, and the headlines were like alien on Earth or something. I go, okay, like this, it's finally happening, but it's just actually a result of poor methodology. So the importance of this case study is there's this microbe that was found that seemed to function off of arsenic. So it questioned the fact that phosphorus is necessary for cellular function. And this is a really huge, it's a paradigm shifting result. So there are a few experiments that I'm going to talk about that relate to this. The first one is, so Wolf Simon et al., they found this microbe, and a few specific claims were made, and I'm going to go into some of the measurements, is this microbe uses arsenate as a nutrient. Okay, so that was the first one. And the second one is it incorporates arsenic into its DNA backbone. That was the important part. And this was, again, a revolutionary discovery, but people in the community started questioning this, and so I'm going to go through two other experiments. The results seem to be robust. Okay, so number one, there are two types of robustness going on. The first one is the strength of the actual organism. So it seems like this organism thrives, at least to a certain extent, 60% of its growth rate in arsenate. So that was the robustness of the organism, but also there was robust measurement that was going on. So there were multiple techniques that were used in order to figure out if this organism was actually incorporating arsenic into its DNA backbone. But other scientists started asking questions, and so a different set of experiments started, and the second set had to do with something a little bit different, which was analyzing phosphate-binding proteins. So this wasn't exactly a replication. In fact, it was kind of coming at the phenomenon from a different vantage point. So the structure of these experiments, and I'm just going to go through this briefly because experiment 3 is the important one. You have the measurement setup. You expose this bacteria to different types of arsenic and also phosphate. And what you find is this bacteria has a high preference, a super high preference for phosphate. And the mechanism, the reason why this is going on is because of the angles with the proteins. So what you see is this bacteria has a preference for phosphate, but now the question still is, can it survive, at least minimally, in an environment where there is no phosphate? So we have to get into some questions about the initial study. So here are some questions. The first one is, and this is a very basic one, when you are working with a sample, how do you handle that sample? So do you purify the sample? Did they wash away any kinds of molecules? And think about it like this. You get residue on your shoes and on all sorts of things. That doesn't mean that these things are tightly bound to these structures. So you have to figure out what kind of bonds are going on. Are they strong covalent bonds? The second one, were the bacteria fed salts that were contaminated with tiny amounts of phosphate? In fact, they admit to this, they were. So perhaps that is influencing the survival of these alien bacteria. Experiment 3, so the findings first. Arsenate does not contribute to growth, and this is when phosphate is limiting. And I'm going to tell you all something in a bit about the sample that was used, because that's probably the most amazing part of this replication. Number 2, DNA purified show no detectable amounts of arsenic in the backbone. And this was something that was very important because they got the sample from the original experimenters. And that makes it so it's a direct replication, but it's a more careful measurement. What they did was they washed away the arsenic, so it was just free arsenic. So the important thing for us, we don't care about this case study aside from the fact that it tells us something methodologically. So the divergence in results indicates something very important, which is this is an experimental error. There was something that happened between the first experiment and the third experiment that methodologically we can make the inference that there's an error. Number 2, the original arsenic consumption effect was an artifact of the lack of the purification in the preparatory procedure. Similar thing to the mesosome. So we see that we have to take care not only in the outcome measurement, but also in the steps like the preparatory procedure. So, in social measurement, do your populations expect certain experiments to happen? The preparatory procedures are just as important as the main measures that are going on. So let's abstract the methodology. Number 1, we have diverging results. So we have a comparison between divergence and convergence, and this draws our attention to something going on. So what you're doing when you're comparing multiple modes, this is kind of like when you ask your friends, like, you ask your friends for life advice, which you should never do, and you go, what's happening with my relationships? And they go, I'll tell you what, and everyone wants to tell you. And the whole thing, and then you end up figuring out that they somehow converge, on that you're messing up your life. So basically, what's going on is we use convergence and divergence to maybe tell us about something that is reliable, but we also use it to tell us when something is malfunctioning. But then we also need theoretical characterization. So theory has to step in and really locate where the error is going on, why the error is going on, how the error is going on. And it doesn't have to be a well-developed theory. It can be a theory about instrumentation. It can be a theory about purifying samples. It can be a very basic type of theory. So M1 and M2 indicate the presence of artifacts. So the thing about robustness analysis is that a lot of examples in robustness analysis actually share something. And I'm going to go through, just list some of these out. So you have signal detection, ecological populations, predator-prey relations, climate change. There's a whole bunch of literature on this also. All of these are about natural phenomena. Okay, so all of these are about something that occurs that you perhaps passively observe. Perhaps you can use the analogy of noise detection. So you get a strong enough signal and all the noise kind of cancels out, and the signal is the thing that you home in on. That's the thing that you're looking for. So perhaps these are certain kinds of measurement objects that we're looking at. So let's see if we can characterize phenomena really quickly, because I think this is kind of relevant to the previous discussion. So the most basic characterization that I'm just going to offer is that you have objects, events, and processes that are discovered. Okay, so these are discovered. They exist independent of these processes, obviously, the measurement processes. but you discover them by comparing multiple independent methods of identification. Bos von Frosten has a nice distinction between appearances and phenomena, where appearances are contents of our scientific activities, measurements, and phenomena are things that exist independent of us. So when we think about natural phenomena, we think about discovering things. And that applies in the bacteria arsenic case, that applies in the mesosome case. It seems to fit very well. But in other cases, it might not. So again, the signal detection analogy, you're going to fine-tune your instrumentation, also your modeling. Perhaps you're fine-tuning your mathematical formalism, and you are somehow discovering something about nature. So the puzzle of variant results, and this is something that I started thinking about because of the biological case study that I worked with, and it was very puzzling because I couldn't get an answer for this, and I couldn't get something that was consistent. It kept on changing at all times, so I'll run you through the example. Much of biological research requires manipulating local experimental conditions. So what you might have is certain regularities only exist within the presence of given measurement setups. And this is problematic because on M1 and M2, these would be classified as non-robust and perhaps unreliable things that you are producing. So when you have robustness, you want consistency, you want invariance, and perhaps these effects will not allow for that. So here's an example, and I'll run through this in very simple terms, and you all can ask more questions. When you're studying turtle populations, which are great, sex is determined by temperature dynamics during incubation. It's not determined by genotype, which is a really, really cool thing. So it's kind of like depending on what temperature it is, and you're a fetus, and your sex is determined a completely different way. So let me just break down a couple of points about TSD. So sex is determined permanently after conception by incubation temperature. There's a pivotal temperature. So if you go below or above the pivotal temperature, you get different dynamics. 3 different profiles for sex determination. And this is probably the most interesting part of TSD, which is there's a certain period at which you are sensitive to temperature effects. This is called the thermosensitivity period. It is the window at which you are developmentally sensitive to temperature effects. So they have measurement setups that occur in the lab, in semi-natural conditions, in natural conditions, and these all produce slightly different results when it comes to something like the thermosensitivity period. So under constant...
What L means?
I'm sorry?
You have male, female, male, and female. What's the L mean, the number 3?
Good question. I don't remember. Let me look that up.
No, that's okay.
I know what that stands for in different terms, but not... I'm going to have to figure that out. I'll get back to you on that one. Good question. Okay. Excuse me. Pardon that. Can we cut that out? You edit that? Okay. Good. So if you have constant temperature conditions which are in the lab, what you end up getting is a very predictable thermosensitivity period. It's right in the middle. It doesn't skew. It doesn't stretch out. Oh, here it is. And this is just a basic characterization of it. But when you start experimenting under field conditions, you see that the thermosensitivity period actually moves along depending on the temperature dynamics. And so you get this disagreement between what's going on within the lab and within the field. And it also matters what kind of field measurement you have. So are the scientists somehow involved in these semi-natural measurements, or are they just kind of laying low? The other thing that matters is turtles are also doing things that are could be regarded as manipulation. So turtles are burying eggs deeper or shallower, and they are also moving agents. Perhaps that matters to the practice also. So you have a very general picture that occurs within setups. Under what conditions are temperature-sensitive developmental phenomena produced? We're not just talking about the thermosensitivity period. We're talking about the sex of the turtle. We're talking about immune function. There's also this thing called the righting response, which is when the turtle is on its back and it flips over. The righting response is even determined differently within these different measurement setups. So depending on if you're doing the measurement in the lab, you get different immune function, righting response, thermosensitivity period. And this matters because between setups, scientists are trying to figure out what to do about global warming. Completely different predictions about what's going to happen with turtle populations between these setups. So the question is, well, you have these unstable measurements, inconsistent measurements, contradictory measurements. how do you compare them? What do you say about reliability and error? Do you say that one of those types of measurements is an artifact or producing many artifacts? So the measurement setups you have in natural conditions, you have in the lab conditions, and now you have in computer simulations, and I drew those out. So that's part of like a little weird animation I did. So in the computer simulation conditions, what's interesting is, and I've boxed out the laboratory conditions, scientists have in a sense given up on the laboratory conditions, and what they've done is they've actually created computer simulations that mimic field conditions, and now they use the computer simulations along with field conditions in order to say that these results are robust. So they have in a sense, and literally in certain cases, they bury the results, so they ignore certain results. I have another paper about this called The Measurement Problem of Turtle Mechanics, and it's a really, really tough one because you have these results that are just ignored some of the time, results that are tossed out, and so now what's going on, like I said, lab measurements are thrown out. So let's apply M1 and M2. So diverging results indicate that TSD measurement in certain conditions, namely the laboratory, produce artifacts. And scientists are actually in agreement, for the most part, that the lab conditions should be tossed out. But I'm not so sure about this. So one of the examples that I've just kind of chosen, and there's a complex and thorny pragmatic aspect that is going on here, is you have this one paper that calls constant temperature lab setup proxies and claim that the use of proxies has been refuted in TSD literature. But the problem is the paper that they're citing is not about refutation at all. The paper that they're citing is a paper where it's just presented that there is this contradiction between field and lab measurement. So what they're doing is they are for some reason choosing this. I mean, the intuition seems natural. The field conditions are the natural conditions, so why would you choose the lab conditions as well? But there's something important to be learned about the lab conditions, I think. And this is why I think that we need a new classificatory system for these types of sensitive phenomena or unstable phenomena. So the final thing that I'm going to talk about is phenomena versus effects, and I think hacking is the best place for this. So hacking characterizes phenomena as observable regularities. These are things when you're looking up at the stars, you're looking at the planets, you are not manipulating the planets and the stars. Sure, you have a certain vantage point, but you're not physically manipulating those things. So a nice quote that summarizes the difference, and I've just bolded the important part, it has to do with intervention. So when you're talking about phenomena, you're talking about events that can be recorded by the gifted observer who does not intervene in the world. So it's a passive measurement that is going on. And when you're talking about effects, you are talking about intervening in the course of nature. And the question that was brought up earlier was a really, really good one, because when you think about the fact that perception is actually active rather than just passive, that there is certain manipulation that is going on even when you're passively observing a population of animals, maybe this is on a continuum. Perhaps the main thing that's going on here is you're talking about physical manipulation with the object under measurement. So effects, according to hacking, experiments create effects, and the aim is to refine, to create, and I really like this idea of reproducing effects. So you want repeatable, reproducible results. And because effects fall apart, it is unlikely that these things are robust. That's kind of where the puzzle comes in. So when you have temperature-dependent sex determination, it seems like you were dealing with an effect. Now, it's natural for us to think about natural setups as the primary ones. But what's really going on here is we are studying different relations within the setups, or perhaps different interactions. So I just want to finish up with context-sensitive effects. I think that we can start thinking about effects in a way that highlights the relational aspect of them. So when we think about phenomena, we don't think about relations. There's all this philosophy of science literature on perspective and representation. Those are kind of passive relationships that we have. But when you have an active relationship, intervention, manipulation, if you look at Woodward also, it's a great source. What you're talking about is the relation between the measurement setup and the object under study. And this is where, this obviously relates to work that's going on in philosophy of quantum mechanics. People like Barad talk about intra-actions rather than interactions, so you have entangled wholes of things rather than just separate objects. So here's the apparatus and here's the object. And this applies in biology also. Sometimes the organism itself is used as an apparatus and as an object at the same time. In social measurement, you are measuring people that are measuring you while you're measuring them. So there are a lot of really cool relations that are going on, and these are not just passive relations, passive perspective. So the other thing is, and this is the thing that I think is really important, the information that is useful out of this is it tells you something about the production process. What we should be doing is not tossing out the lab results, but we should be figuring out how certain effects are being produced. What is the relationship between manipulating certain measurement setup conditions and the object under study? So we should be asking how questions. And this is an independent shout out to Alok. I have this distinction, I did not see yours. I think there's a distinction between taking a measurement versus making a measurement. And I think that what we're doing when we're dealing with context-sensitive effects is actually making measurements. And this is not just reserved in biology, so I want to finish up with a physical example. Chang talks about the boiling point of water, and he talks about fixed temperature points. And when you think about the boiling point of water, it seems like a passive sort of measurement. What you're doing is putting a thermometer into a sample, and the boiling point of water comes out consistent every time. And this is just something that we discover in nature. But actually, there are all these conditions that the boiling point of water depends on: atmospheric pressure, dissolved gas. All of these conditions have to be stabilized, in a sense have to be, well, according to Chang, we manufacture these conditions. And these conditions, once we manufacture them, we can have a repeatable, reproducible sample of boiling point. And then once we've standardized that, we go, simple, we've discovered this. So we kind of abstract out the whole intervention story. But the intervention story is important. So I'll finish up with a final thought, and this is kind of a puzzle about unstable effects. Effects are informative. They teach us about the production relation. They make us take a slightly different vantage point on the objects under study. They're no longer things that we passively observe and discover. They are things that are relational. But here's a problem. If effects require careful experimental production and are sensitive to change, we still have to answer this question at a certain point. How do we know when an effect is genuine rather than when it is a result in error? And this requires a slightly different characterization of genuine. No longer are we talking about things that are just natural. So I talk about this a little bit, and I think that this has to do with pragmatic as well as theoretical considerations. At a certain point, we have to figure out when it's enough to do a certain preparatory procedure, or, one of my favorite examples is we always talk about the independence of the senses, but the senses aren't really independent. They have one compromised systematic flaw, which is the brain that processes all the stimulus. And in those cases, we still go, the senses are independent enough. So we have certain pragmatic considerations that we use, and I think that that's where we can perhaps differentiate things that are genuine from things that are produced in error. Thanks a lot. Yep.
So that is great. That's
Thank you.
It's very interesting.
You missed the initial,
No, but I
bank robber.
came in with the introduction, and
Okay.
I got most of it. So I have a copy of the latest Scientist with me. I read on the plane, and it's about the reproducibility problem in science. Massive.
Yeah.
The article makes this big claim about, essentially everything is an artifact. Most of it is an artifact. Artifacts, right?
Yeah.
That's
And in
the
social,
definition.
is it mostly about social sciences?
It's actually about science as in the
Wow.
right, yeah. I'm talking about publication practices and all the things that we know about already, how it adds up to the story of how reproducibility is really an issue right now in science. And the problem is, so the article makes the point that people should be updating their own articles as time. It shouldn't be a static document. You publish it and you're gone. You should
Right.
update it as new results come out, because most of it is probably untrue when you dig into the academic literature. Anyway, that's a side point.
Mm-hmm.
If that is true to some extent, how can artifacts then be classified as kinds? maybe they are non-kinds. Because if they don't actually produce a genuine effect, how can we classify them as being a thing?
So there are maybe two cool issues going on here. One is that genuine effects are unstable. So, and I the reason why I asked about social science is because this came out recently also, that all these classic social science experiments are not being replicated. And the reason why is because the thing, the phenomenon is changing, perhaps the measurement setup is changing. So you're failing to replicate the genuine effects, but maybe you also fail to replicate the artifacts. And so if both of these things, you fail to replicate both of them, how can you say it's a kind? Maybe it's like it's a moving kind, or like I say, I think it's, the way that I always think about things is in terms of measurement setups. So it comes as a whole. And when you tweak, certain aspects, you change the thing that you were actually measuring. So I think they're changeable kinds, or something like that, like dynamic kinds, something like that. Yeah.
There was a book in the 1980s or so that was talking about the reproducibility measurement, which I can send you the reference to, but that last point you just made, they say they tried to reproduce 3 types of experiments. One was a plasma physics experiment, and when they went back to reproduce it, the signal analyzer had changed. The old one had broken, and the manufacturer didn't have the same model. They sent a new one, and it was a little bit more sensitive, virtually impossible. And it really sensitized me to some of these issues about what does reproducibility in science really mean. But I want to go back to your general point, which I could not agree with more. And I'm going to ask a slightly different question than you think, is how can you communicate this to scientists in a way that they understand, their fallibility? Because I find that there's a human aspect of science that people want to see something, so they see it. And they create, especially in experimental and modeling situations, you make assumptions about what's important. And those assumptions create those conditions that produce unstable effects. Mm-hmm. And it takes a lot often to persuade people that they're making a lot of implicit assumptions that they don't articulate, and they'll deny that they're making, and it really affects scientific results. And you have, and the more distinguished a scientist is, unfortunately, the more
right.
the stronger they're convinced they're right. That's not totally true, because maybe the really great scientists understand their limitations. Yeah. I mean, have you tried to provide feedback to, say, the turtle measurement community or other communities to say, look, think about these things, and what kind of reaction have you had when you provided that feedback?
Mostly like, thanks, and then that's it. But yeah, well, I think two things there. One is people like stability. So whether it's engineering something and getting a repeatable result, or finding something and having this realist attitude towards it. You see this a lot in physics, but also in biology. You don't want to say natural selection is somehow an effect or something like that, but it is sometimes certain kinds of interactions. So I think that that's one of the big things is people like stability. The second thing is there's a lot of grant money riding on this stuff, and so if you go, hey, don't drop the lab setups, and they just if they had a landline, they would just hang up on me because it doesn't matter. So communicating with scientists is obviously a tough thing. This was one of the coolest experiences I had, because usually at philosophy conferences you have it's like really aggressive. It's like an actual fight. And then I went to the American Geophysical Union, and I presented on climate models. And I was expecting to be in a brawl, because I'm like one of the only 2 philosophers of science. There are thousands of climate scientists. I was like, this is going to be bad. And they were so supportive. They're like, this is really great stuff. And I was not used to it at all. So in that area, biologists are also pretty cool when it comes to talking about the methodological aspect. They appreciate, because they do agree that phenomena are more movable and dynamic than, perhaps classic philosophers of science think. So I guess maybe it depends on the population of scientists. Good question, though. Yeah.
Yeah, it seems like that was a great talk, by the way.
Thanks.
It seems like the context sensitivity problem with robustness, I mean, it doesn't even depend on interactivity. It seems like it sort of transcends that even, because I can think of a lot of physics experiments where somebody measured something and nothing else was sensitive enough to see that. And so basically the whole topic got dropped. And so it really affects the questions you ask even sometimes.
Maybe they're also, one of the questions I think about is sometimes you get a constant and you wonder, is that constant something that is robust, or is it something that is foundational for the practice or the set of models to work? It's almost like you have to have this in place. It's a necessity. So is it something that is just, I forget the distinction that I make, but it's something that's just repeatable, but it's not reliable, basically. Yeah.
I guess our NIST representative kind of.
Yeah. Saw some hands. Mm-hmm.
Yeah, this is great. When, if ever, do we get to stop calling something context sensitive? Because in one sense, like, all of science is a context.
Yeah.
Do you have a sense? I mean, and not like a hard and fast, these are my definitional terms, but even just some sort of intuition or methodological, like, this is when we call something context sensitive versus robust.
Well, it's interesting, so the reason why I specify there are things that are context sensitive and then I say maybe there are things that are not is because I really got a lot of resistance from old school philosophers of science on this, because they really want to say that there are certain phenomena, and this, the classic example is, you look at the stars, you're not intervening in any sense, but there's maybe an inertial frame of reference that you have to talk about. Maybe there's something that's important, outside of, we talked about aliens earlier, and all I could think about was Slaughterhouse-5 with the Tralfalmadorians. All time is all time. So they have a completely different frame of reference from us. So I think that, to be honest with you, and we can shut the film off, I would think that all phenomena are context sensitive, but that would be very controversial. That would be really controversial.
So what about the context sensitivity then of not just the phenomenon, but the measurement? Mm-hmm. So I record my data, and I can choose to annotate that data with a lot of really specific information about the apparatus that I use to do that measurement, or not. It seems like adding that sort of annotation is specifying, the context of that measurement and that number itself. So the effect may be a function of a particular measurement setup, but the specific measurement itself, the number might be as well.
Is also context sensitive. And you're also, you're not just working with raw data. What you're doing is smoothing out the data, making data models, and now we have, big data is a big thing. And so we talked about this a couple weeks ago with some other people in this room, but big data is not just, you don't get all the data at once and it's, just there for the taking. You have to categorize things, you have to move things around. And when you do that, perhaps you're making things context sensitive.
I tried to show a slide of some of this that say, okay, there are 4 or 5 different ways you gather data. One is observation, one is experimental, one is computation, one is doing surveys, one is testing. And what do you do when you do an experiment? You try to create an isolated section of nature and control everything in that nature. And almost by definition, every experiment is going to be context sensitive. What you really ultimately are trying to do is to make sure that you know everything that really should be controlled, and at that point, hopefully you generate what you asked me later on, or earlier about properties. rather than just a measurement result. As that guy Chain said, the boiling, determining the boiling point of water, you eliminate all the context-sensitive, independent variables. You've controlled them all. For observations, what gets interesting is while the phenomena or whatever you're looking at is natural, you're looking at it in a context-sensitive way. That every instrument has some context associated with it. And you mentioned looking at the stars. It took people a long time to realize that if you look at the stars through the atmosphere, you're not seeing, the stars. Mm-hmm. And then it took them a long time to realize that the light that's traveling to you goes through dark matter, cloud, all sorts of things. And so, what you think you're observing, you're not really observing. And that's just a process of science to eliminate those independent variables, or at least understand them and eliminate their, the lack of control of them. And I think you have some really good examples of what happens when you don't do that.
Yeah, the way that I characterize it is you want repeatable, reproducible results. Science gets by.
It could be reproducibly wrong.
Right. Like the fast neutrinos, right? That's good stuff.
Yeah.
Yeah. Well, and that's and you fail, and then you keep on trying, but it's almost like the pragmatic aim. That's what we really like. We have mastered making thin film transistors, and now we can use them in our TVs. Cool. Like that's it works well for us. And so we like the engineering aspect, but we also like observing the same thing under certain conditions. But the big thing here, it's still going to be attention. Maybe we can talk about this at dinner or wherever. But this tension between perspective versus engineering versus manufacturing is really a big one. And I think you get this tension between classic philosophy of science and stuff that's going on in Phil Bio right now, or perhaps history of biology, history of science. People talk more about engineering and manipulation. Let's go right here, and then we'll get the grouch.
So there's one piece I also want to throw on the table. I think this is an important piece, and that is when you actively create the artifact, and that's the reporting problem. So you can do all the science.
Yeah.
If there's experimental error, that's not necessarily your fault.
Yeah.
But when you actively create an artifact, and the part of the I can never say that word. Our problem is where people, first of all, will publish only the positive results, don't publish the negative, and there's all these things that are coming out about reporting, and all the biases associated with it, and the reasons why people report one way or the other. They're not actively trying to deceive, but they're reporting in a certain way.
Right.
So there's that, so you're actually actively creating those artifacts, which go out there and people rely on them and continue.
That's how people think about artifacts in philosophy of technology.
Okay.
So these are things that there's an intention component perhaps, or it's characterized a little bit differently in that field, but you're actively pursuing these things. Or when people talk about artifacts like in art, for example, or they carry a certain significance or whatever. So you might be actively trying to create certain artifacts. In science, maybe you're doing it just to see what kind of errors you can produce so you can avoid them. Maybe that's a smart strategy. No. Yes.
Sorry?
Go ahead. Yeah.
In your talk and to some extent in the questions and comments afterwards, there's been a strong tendency to equate error and artifact. And I think in recent decades there's been a tendency to distinguish between various kinds of anomaly, for want of a better term.
Yeah.
One of which is artifacts, as is perfectly exemplified by the fast neutrinos kind of thing. And we knew it was an artifact because we have this theory, and we know that the theory is absolutely wrong, right, and infallible, and therefore when we get results that are contradict theory, there must be an artifact. So we go find the artifact. That's different from a random error,
Right.
Random errors are immediately identified by robustness types of techniques because you can't get the same result.
Right.
Whereas in some cases, you do get the same
Right.
result.
Systematic versus random error.
So what you're looking for is, on one side, accidental random error, and on the other hand, what Bill Wimsatt called bias when he wrote those papers back in the 80s. And those are two different kinds of things, and they have to be detected by the use of robustness techniques, but different robustness techniques.
That's right. Yeah, and the independence is
furthermore,
important for that. Yeah, go ahead.
You have to additionally recognize the existence of two more kinds of anomaly. One, you might actually discover something that nobody knew about before, all right? It happens.
Yeah.
In comparative biology, it happens a lot.
Like a Sasquatch.
Yeah. Or a new species of beetle.
Yeah. Cool.
And the fourth one is, I know this is going to come as a terrible shock. Don't quote me outside this room. Sometimes scientists fake data.
Yeah, I mean that is.
And that's a logical possibility which, under elderly circumstances, you need to be able to rule out.
Yeah, that's good. So random error, systematic error,
Type 1, Type 2 uncertainty.
Bias, new discoveries, and then just scientists.
Well, ISO has a terminology for that, Type 1, Type 2 uncertainty.
I'm sorry?
The third one, if you want to,
Who has the terminology?
ISO, the Guide to Uncertainty and Measurement, GUM,
Yeah.
it's been,
It's
how it governs.
I'll
how
email you a
people
copy. They got, what is
report
it,
on
2008 or 2011 now? I don't remember. They
Well,
updated
it came out in.
It's a great read. It's very boring.
And then the third type, if you want to study one which is something new, the discovery of squid detectors was exactly that. And
Of what?
squid, these superconducting detectors, and these guys found a little thing, little anomaly, and they went to the boss and said, we don't understand that, and this was back in the 50 years ago. They said, why don't you spend a lifetime figuring it out? They won Nobel Prizes, and there's a whole industry associated with it, but it grew from what your third thing, which was something, an anomaly that was real.
Yeah.
It took a long time to
That's
figure
good.
out.
It's a really good distinction.
I'll send it.
So maybe we can continue some of these conversations over dinner.
Thanks for the comments, y'all. Good.